go, great. So, thank you very much. I was very nervous in the cab driver, but in the cab I tipped the driver extra special money for getting here on time, so that's very, very lucky. My name is Simon Gane. I'm an ENT Registrar, not quite a grown-up ENT surgeon like Carl, but not far off there. And so what is it, ENT? Ear, nose and throat surgery, as you imagine, it's one of those very obvious surgical names. We generally do ear, noses and throats. But I always talk about it as being really the surgery of the senses, because apart from vision, well, actually we do things with the orbit and around the oculoplastics on the eye, but we don't touch vision. But apart from that, the other 5 canonical senses, we all have something to do with. Certainly hearing and ears, ENT surgeons have a lot to do with. Smell, obviously, otherwise I wouldn't be here today. Taste, for the same reason, we have a lot of people who have mouth problems, and we share that with most of the facial surgeons. And then touch, well, you can't as a surgeon really avoid the sensation of touch and pain in your day-to-day job. It's just part of the package, unfortunately. And even the other non-canonical senses, things like proprioception and balance, we also have a lot to do with. It's all part and parcel of the thing. So if anybody's going to be talking to you about smell and taste and indeed all of the senses, then it should be really an ENT surgeon. So that's me. And what I want to do is to talk about what you're here for today in terms of those senses. Because what we think of as taste and smell and touch, they all interact with each other to form what we call chemoreception, chemosensation, chemoperception, in a sort of overlapping way. So that overlap is really what we're interested in today, because if, for instance, you can't smell, those other 2 things become more important. And if you know that, you can take steps to address that, to make food more interesting, for instance, or to increase your perception of the environment in those ways. So that overlap is what I'm interested in today, and that's what I hope to be talking about, or I am going to be talking about for the rest of the day. So let's call that chemosensation. So my specialist interest, I'm completing a PhD in smell, in the physics of smell, which is kind of weird. I'm interested in the olfactory receptor, which I'm going to tell you about in a little bit, and how it recognizes molecules. And we think that there may be a weird quantum phenomenon going on in that recognition step. That's not why I'm here today, so shall I do more about that? But what I thought I'd do is take the sensation of smell and use that as a canonical example of how this receptor-based sensations work. And you can apply that then to taste and to touch, and as a sort of an example of how all of them work. Also, I know most about smell. I don't really know much about taste and touch, so that's why I'm going to be talking about smell. So William Shakespeare famously said, "A rose by any other name, the smell is sweet." So here's our rose. What happens when you stick a rose in a gas chromatograph and you see what's coming out of that? What makes the sense of a rose? But actually, if you start analysing it, there's just all these chemicals coming off the... I cheated a bit there. That's actually a duplicate of this. But there's a lot. There's a mix of chemicals. That's just from a rose. You can imagine all the sensations smell. There are multiple chemicals getting into the nose all the time. And how does that translate into the sensation of the smell of a rose? Well, it's a mess, as you can say. So let's just take one of the components, benzaldehyde, one of my favourites. It smells of marzipan, actually, almonds. And it looks like this. Bizarrely, if you stick a little 3 carbons on this side, it changes the smell completely to one of cumin, called cuminaldehyde. And that's part of why I'm interested in sense of smell. But anyway, benzaldehyde, what it has to do is to get into the nose in some way and then interact with the olfactory receptors, with the olfactory nerve, and cause a signal to then propagate upwards to the brain. And that would be the sense of smell. So how does it do that? Well, let's look inside the nose a bit more. Inside the nose, the blue things are the first cranial nerve, the olfactory nerve, the projection of a lot of nerve fibers into the nose, where they're going to be looking for the molecules that are in there. So each of those nerve fibers is made up of many multiples of the single olfactory sensory neurons. And they look kind of like these funny hydra-headed nerve cells. They've got lots and lots of ciliary projections into the mucus layer that's in this area. And on those cilia, on the cell membrane of those cilia, if we zoom in even further, so that grey thing is still the cell membrane, are lying hundreds, probably, we don't actually know, olfactory receptors. So these receptors are called G protein linked receptors. That's a sort of their general class, and these G protein linked receptors are very interesting from a research point of view because most drugs that we have, apart from steroid drugs, are aimed at receptors just like this. But basically, what they do is they recognize the molecule. So let's see if we can get our molecule up there. That's our benzaldehyde in the nose. Somehow it interacts with the receptor, and that's my research interest. I'm not going to talk about that today. Activates the receptor, so it recognizes it, activates it. Then that activation is then kicked along to something called the G protein, which is sitting on the inside of the cell. That G protein then causes the dissociation and a particular part of it, the alpha subunit, and activates something called the CAPase, which makes a second messenger in the cell, and blah, 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 blah. There are lots and lots of steps. And basically, what happens is it changes the electrical balance of the whole cell. And that's what we call depolarization and activation. So it changes the electrical balance, and so we can represent that as now red through various ways. And if you do this enough, instead of just changing the balance of the local area, you can depolarize the whole cell, hopefully, and then that starts to send a signal back up the olfactory sensory neuron, up the nerve, into a part of the brain called the olfactory bulb. That's that blob there. And then something interesting happens. So that you thought that was complicated, it gets worse. The bulb is the next representation. The bulb is very clever because the problem with these olfactory sensory neurons is they're constantly dying off. These are nerve cells that are directly exposed to the environment. They're dying off in their hundreds all the time. And if you're a brain, you want to recognize, you want to have some wiring in the brain that's more or less stable. And if you're dealing with hundreds of nerve cells dying off all the time, what you need to do is to have some sort of fixed wiring and then a clever way of getting the dying off cells to go to the right place. And this is what happens in the bulb. So let's say an olfactory sensory neuron recognizes an M, for instance. Just let's say completely hypothetical. We can call that the M olfactory sensory neuron. And then millions of those in the nose, and they all recognize M's. They, through something that we don't quite understand at the moment, when they're growing from the basement layer where they start, they can find their way, all of their way, to a particular part of the bulb called the glomerulus. So that area, that glomerulus, we might as well call the M glomerulus, right? So all the neurons bearing that receptor, which there are 400 different types, go to the M glomerulus. So then you say, well, what about E? Well, let's call that the E glomerulus. And then what the brain does is it can recognize the pattern of activation of these different types of glomeruli as a smell. So it's the pattern of activation. But the clever thing is, these things can recognize other smells. So it may be that the O and the T are activated, but instead of the M and the others being activated, you activate a P one. You get a different type of cell. So what I'm saying is that the pattern of activation, even though there may be overlap, that's what's recognized as a smell by your brain. And that's how very cleverly the brain deals with this problem of constantly regenerating olfactory sensory neurons. Of course, that's one of the reasons that we can get anosmia is that regrowth is disrupted in some way. Carl will talk about that more later. So that's the bulb, and that's really, I'm not going to talk anything about the higher centers and how that's all integrated until later on. But that's really smell. So that is all mediated by the first cranial nerve. So there are 12 pairs of cranial nerves. These are direct extensions of the brain. Everything else, every other nerve in your body goes via the spinal cord apart from these 12, which are direct extensions of the brain called the cranial nerves. The first cranial nerve is the olfactory nerve that we just talked about up there. And it gets data from 2 points. It gets orthonasal information, so there's molecules coming up when you sniff for those. But for instance, if you ate something that was rose flavoured, like Turkish delight, for instance, it would also have something called retronasal olfaction. A large part of what most people call taste is actually smell, which I'm sure we're going to go on and on about today. And this is this retronasal olfactory path via the connections at the back of the nose to the posterior point. So that's pure smell. But there are, as I said, 400 different types of these receptors, so it gets very complex very quickly. So you might get one pattern of activation that gives you that particular smell, another one that gives you that particular smell. And if you look at that 400 times itself, there's about, that's at least 160,000 different smells that you might be able to recognize. And that's just assuming that the receptors have an on-off state. They probably don't. They're probably more analog than that. You can have a more activated or less activated sensory neuron. So it's very, very complex. The other ones aren't, the other modalities that I'm going to talk about aren't that complex, but they all feed together to make a really complex process. So that's smell.